variant associated with autism.
Thanks, Alexander. Thanks, Kathleen. Thanks, Caroline, for this amazing talk about everything I will say is something that will follow what you said. I'm from Institut Pasteur, man. Not Curie, but it's no problem. Yeah, sorry. Okay, so I spent all my life almost, sorry. Oops. Are you goodbye? Yeah, so how do you go there? Okay, so sorry. I spent all my life to find genes for going that way. And so I'm trying now to get all my life to find some resilience factors and not risk factors anymore. And what is red and what is green has to be also discussed, I think. So the first questions, this is an education session. Are there genes in the human brain? So if you live in France, sometimes you think it's a little bit like this. That genes can be, it's, you know, there is no question that there is genes for kidney and for heart or liver, but for brain, it's always complex. And it's not a new thing. I mean, the guy on the left is Charles Darwin. And once he received the visit of a young person, Alfred Wallace, who said, I think I found something, I think the natural selection. And Charles Darwin was very shocked because that was his theory, but he never published it. And so Wallace said, okay, this is your theory, and I'm a Darwinian. And, but then later he published a little paper saying that for the brain, it's very strange because the brain can do things that are not selected yet. So maybe natural selection, I don't know if it exists in the brain, blah, blah, blah. And Charles Darwin sent an email to Alfred Wallace, and he said, I hope you have not murdered too completely your own and my child. So you see that the brain is always complex from the very beginning. So I work at the Institut Pasteur, but the door of my lab is really at the hospital, and I work with amazing clinicians who see the people with autism, their parents, but they're also their undiagnosed brothers and sisters. And the data are coming to the lab, and so you have genetics, brain imaging. Sometimes we found a gene and we can make a mouse. And so this is in Paris, but we also are working on at the European level. And the last project that I coordinate is called R2D2, Mental Health. So R2D2 stands for Risk, Resilience, and Developmental Diversity. And in that project, we want to understand resilience, and we are working with some people that are super expert in autism, who are people with autism. And I think that we lost this idea of trying to work more as a participatory research. And so they really shed light on what autism is. So we call it Autism's Plus. Because yes, autism is quite simple. It's atypicalities in social communication, stereotypy, restrictive pattern of interest. But it's very, very diverse. So some people still think that there is a prototypical autism. But most of the time, autism doesn't come in an isolation. It's coming with intellectual disability, epilepsy, sleep problem, sensory motor. This is something that we didn't really look at. And when you listen to people with autism, this is everyday life. This is one of the main problems. Social interaction is not a problem, maybe, but sensory motor problem is a problem. So this is the plus. And we know, and this is exactly what Caroline said, that there is a continuum. So who is really autistic, who has autistic traits, the boundaries are not so easy. We know that there are more males than females in autism, but the brief answer is I don't know why. But I know we are very bad in diagnosing girls with autism. And the prevalence is about 1 to 2%. And it was not the case a long time ago, so it was super rare in the population. And so it goes like this, and I think in California now it's 1 in 22. So why? So because autism is really not the same as from the beginning. The diagnostic criteria are much larger, and also I had the chance to meet Stephen Shore. He's a person with autism, and he wrote the book Autism for Dummies, which is really a great book. And I was in New York City, and he said it's easy that the number of people with autism increase exactly the same as the number of FedEx package in the world. And I was a little bit jet lagged, and he said, of course, there is probably no association between the FedEx package and autism. But then he asked us, can you see what is between the E and the X in FedEx? Okay, so I was jet lagged. I didn't get it at all. But then I realized that there is an arrow. Oops, sorry. Oops. Can you see the arrow now? So you have learned something today. So every time you will see a FedEx package, you will see the arrow, and you will think of me and autism. So, and he said, autism is a little bit like the arrow of FedEx. Now that you can see it, you can't miss it. And so that's exactly that. We are much more aware about autism now. And this is a really beautiful work by Santosh. Normally I give that talk. Santosh is not in the room, but today is in the room, so you can go to see him. What he has shown, it's really beautiful. He showed that, yes, there are more people with autism, but there is also less people with intellectual disability. So most of the time now, when you have a child who is coming to the clinic and he has intellectual disability, but now you look at the social interaction problems, you look at the stereotypes, and you have a diagnosis of autism. And also, I don't know how it is in Germany, but in US, sometimes in some states, maybe all states, if you have a diagnosis of autism, if your child has a diagnosis of autism, you have more chance to get medical care and support than if your child has intellectual disability. And it's the same in France, and I think it's totally unethical, but this is, and that's why some medical doctors are diagnosing autism, that maybe it's not super autism. Okay. So irritability is super high, more than 87%, and yeah, it's probably, I mean, most of the time it's not like this, with the papa like this, the maman like this, and the child like this. But sometimes a single gene, a single variant can make a difference. And this was these genes that we found 20 years ago, and that they pointed at the synapse. So a single variant here, an extra T making a stop. It was an X-linked condition, and these neural ligands at the synapse were missing, and you have autism. And this is, for example, this boy. Yeah, he has autism, not a super severe intellectual disability, very typical autism. His brother, he had Asperger's syndrome. He wanted to be a nurse, then he wanted to be a taxi driver. He knows all the streets of Göteborg where he lives, and he can draw so really the autism without intellectual disability. For Shang-Free, it's a different story. It's a synaptic scaffolding proteins. When you have a mutation, a single mutation, I mean, and you still have one gene, so it's really haploinsufficiency, you have almost no speech, intellectual disability. But, and I was really shocked when I found that. So this little girl, she lost one copy of Shang-Free. She has intellectual disability, lost no speech. And his brother, he has 3 copies of Shang-Free, so 1 extra copy. He started to speak before the normal age. He has a huge vocabulary, and he has, he was diagnosed with Asperger's syndrome at the age of 13 years old. And he's an amazing person, and I met him. Okay, these are the people who are working. The PhD students, the technician, as you can see, they have done other things than finding genes for autism. And Christelle, who was the PhD student who found this Shang-Free gene. So we have this kind of architecture that you have really like de novo mutation and you have autism. Or on the other part of the spectrum, you have probably these polygenic forms of autism where this more than 10,000 variants are accumulating and the child has autism. So for these genes, we have about, 1,800 genes, NDD, maybe 200 genes for autism. I will talk about it. And for the other, we have GWAS. It's a really complex GWAS for autism because it's super heterogeneous. But you also have GWAS for ADHD, GWAS for IQ, which are probably correlated, were correlated. And these are the people, and I think it's for the young generation, you have to choose one day if you're monogenic or polygenic. And so be aware that in the future, you will probably be in the middle to deal with these two kind of variants. So instead of autism, yes or no, we wanted to be a little bit more precise. And there is this test designed by Simon Baron-Cohen called the Read the Mind in the Eye test. So just the eyes, and the idea is to have this cognitive, it's a cognitive empathy. So are you able to detect the emotion of the others? So if I give you the eyes of these guys and say, is it playful, comforting, irritated, bored? So you press here. You have 36 pairs of eyes, and then you have your score. You can do this on our website if you want. So if you have 35, 36, you're super skilled in detecting the emotion in the eyes of the people. If you're there, you're on the average. If you're there, it's okay, but probably you're less skilled. My brother-in-law is there. And it's interesting because he said, I take a lot of information in the mouse. And some people with autism, they take some information in the mouth. But neurotypical, me, I really look at the eyes. That's kind of thing that there is really a diversity in the human population. So we have done our work, and this is Varun Warrier and Simon Baron-Cohen. They asked for more than 700 twins. There is about 28% of heritability. And then we ask 23andMe to put the test online. We get 88,000 participants. So, and the GWAS, the heritability captured by SNPs for the SNP aficionados, it's about 6%. So it's the beginning of this kind of studies. We have done the Read the Mind in the Eye test, but we also have done empathy quotient and systemizing quotient. Systemizing is, do you like to order things, to solve problems, like a little bit the engineer type. So I like this photo of the guy with the little boy who ordered the toys like that. Okay, so if you do that with Varun really made a really great work, you can see that the autism GWAS is positively correlated to systemizing and negatively correlated to this empathy quotient. So it means that instead, again, of autism, yes or no, you can now start to see what are the difficulties in empathy, cognitive empathy, and systemizing. And Jacob found that, for example, that the GWAS of autism was positively correlated to IQ. So I will give you two stories and a bonus track on Schenk. So autism, yes or no, we want to get rid of this, and so we made some factor analysis. Why? Because it's maybe better for specific interventions. So we used the Spark data from 24,000 autistic individuals with the social communication questionnaire and repetitive behavior scale. And so we found these factors which are not super surprising: sensory motor, insistence of sameness, and so on. And so when you have these factors, then you can start to ask simple questions. Are males and females more different? And you can see that most of the time, males, they have a higher score. But for F4, the females have higher scores. And F4 is self-injurious. So I think it's more important to understand why some people have self-injury and instead, I mean, it's complementary to autism. And this is something that we found previously, that self-injury was more frequently seen in girls with autism. So then you can ask also a question about what is the consequence of having this de novo variant. So this is the facet, and as you can see, you have a little bit more intensity of symptoms if you have these variants, but not so much. But what is the major impact is lower UIQ. Okay, and also we looked at developmental milestones, and you have a delay in, for example, the first words. And so this is this kind of high penetrant, large effect variants. The polygenic score are interesting. So if you look at the effect of the polygenic score, so you look at all your autistic individuals and you see what is the symptom. When you have a high score of polygenic score for autism, yeah, you have a little bit of intensity. It's nominally significant, but okay. But then, if you look at the polygenic score for ADHD, attention deficit hyperactivity disorder, you see that you have higher intensity of symptoms. And if you take the polygenic score of educational year, you have lower intensity of the symptoms. So it means that it's really, the background will make a difference in terms of intensity. So in this second paper by Thomas, we looked at loss of function, very similar to what Caroline said before. We took these genes for autism, about 200 genes for autism, and we looked at the loss of function. And so the tour de force was to look at 13,000 autistic individuals, but also to 200,000 people from UKB and the parents and so on. And I am from that generation where we were talking about curation. I asked the people from my life, I could be dead, because I asked them to look at the BAM files of all these loss of function. So that's, so now I can't say that anymore because we have more, but I wanted to be sure that the variance was there. So. And this is thanks to the data sharing from the Spark, from the Safari and the Biobank and the IPSIC. So if you like p-value, you're super happy because you're looking at genes for autism in autism versus controls. And so you have more of this variance in autism than in parents or in controls. But still, you have about 0.5, 1% of people who are not diagnosed with autism who carry this variance. And it's very different from one gene to another. So this is the odd ratio for autism, and this is the number of carriers. So you can see that for these genes here, we never found any carrier in the general population carrying a loss of function mutation in these genes. But for the others, we could find at least 1, 2, 3, 4 of these individuals in the general population who are not diagnosed with autism. We will come back to this. And we carry this variance. So now that you have this odd ratio for autism, you can ask, again, simple questions. Is the odd ratio different when the gene is expressed early or late? And as expected, the odd ratio is higher for genes which are expressed very early during brain development. The second, you can ask, in which module of expression in the brain the odd ratio is higher, and it's more, I mean, you can go to the paper, I will have no time, but it's a little bit more in the neurons than compared to the other cell types that the odd ratio is higher. But I think the most important question was, who are the loss carriers in the undiagnosed population? And so we looked at these 18,000 traits from UKB, and we looked, so if you are a carrier or if you're not a carrier, what are the differences? And in fact, we found that educational score, employment score, health score, income score were popping up. So fluid intelligence, for example, you have, if you're a loss function in the general population compared to the others, you have a little bit less fluid intelligence, similarly to what Caroline showed. You have a little bit less qualification, a little bit less of income, and a little bit more of material deprivation. But still, you have some people who can flourish with these variants. I mean, you can carry these variants of having more money than I have, so which is not super difficult. But. Okay, for the polygenic score, the picture is very, very different. So you have the people who have high polygenic score for autism, they have a little bit more score of intelligence, of IQ, a little bit more qualification, but surprisingly, no difference in income and more material deprivation. So these are my students. I mean, a little bit more clever, a little bit more qualification, but income, boom, and material deprivation. And I think that's a problem. Really, I think that it also shows that there are some people in the society where we can do something and they are maybe they have a problem then. So we have to think about it. Bias everywhere, we know that, and so there was a study where they looked at who is participating. So they made this genetics of participating to the biobank questionnaire. And they found that the people who were participating to the questionnaire, who filled the response, they have higher polygenic score for educational year, for example. So not surprising, we found that these loss of function mutation carriers, they were less likely to go to the MRI scan, less likely respond to the fluid intelligence score, income, or qualification. So, to summarize this part, if you carry this loss of function mutation, sometimes you have a diagnosis, sometimes you don't have a diagnosis, sometimes you're probably in the middle, and also, I mean, the people from UK Biobank, they are, I think, 60 years old now. So I'm sure that if they were born in California now, I mean, some of these people without the diagnosis could have the diagnosis. So this is also important to get an idea of the access of diagnosis. So what makes a difference? And an obvious question that are males and females more resilient to autism? So if you take this model with high burden of de novo mutation or rare mutations and high burden of polygenic score, we looked at this loss of function in the autistic individuals. And we found that there were more girls in the autistic population with these variants, okay, with this loss of function mutation. But surprisingly, when we looked at the population, 50-50, so in the sibling of the autistic individuals, in the parents of the autistic individuals, and the UK Biobank, we found 50-50. So it means that if you have this quite high penetrance variance, you have no difference between male and female. It's like a car accident. It's not a difference. So, and by the way, if you have Sheng-3, for example, mutation, you have equally male and female. And for most of these variants. So the idea is that maybe it's a little bit like this, with, for these variants, no difference between male and female, and for high polygenic score, probably not so much difference between male and female, but maybe males are a little bit more sensitive to a medium burden or medium polygenic score. But then Larnawing came with the first time with this idea of camouflage. So camouflage is that people with autistic traits or that could have an autism diagnosis cope with this and trying to mimic neurotypic. And we know that some girls are doing this. They don't say that they are autistic, and they are trying to get to be neurotypical. And I think that we have to be very careful with that, because William Mendy, he showed beautifully, many studies, that these women were using camouflage to hide their autistic traits. They are more depression than the other autistic individuals. they have more anxiety, and they're really lower quality of life. So we have to really, again, to accept that there is some diversity in the world and that these people can be accepted as autistic, and they don't have to mimic neurotypical. So the paper came out last year, and there was a really nice news and views by Luca and Christine on how can we talk about autism, genetics, and neurodiversity, and I think that's important. Okay, so if you were there last night, you know what it is and I can remember now, about the light alcohol or strong alcohol. And this lady here, I mean, if you go to a concert, if this lady stops singing, you will hear directly the difference. But if this guy stop playing, unless you have a super good ear, it will be difficult. But if these guys stop playing, you will hear the difference. So this is Madame Chang-Free, and this is all the snips everywhere. So if you take a dot here, you will not, you still see the Eiffel Tower if you take out a drop here. But I think this is super important for the young generation to understand that, yes, we can look at this large effect, but these small effects are super strong and super important, okay? So for this very strong effect, now we have a better, this is a protein-protein interaction, and the more the circle is big, the more odd ratio for autism. So now we can have a better, we can have a better idea on what are the genes which are synaptic, chromatin, involved in epilepsy and autism. I will go very fast, but some of these genes which are expressed very early during brain development are chromatin remodeling genes. The others that goes a little bit up are more synaptic genes, and some of the others which are stable or very low, they are a mix of chromatin and synaptic. So we can start to make a better way of looking at the genes that are involved in autism. For the musicians, the SNPs, we know it's not, they are not playing just one song. They are the guys who are playing for autism, they're also playing for ADHD. I mean, but they play a different song for subjective well-being. So that's kind of thing that we have to understand the color of your genome. So we made this GeneTrack website. You can go to GeneTrack if you want to see if your gene is involved in autism. You put your favorite gene. It will tell you if this is a gene that is in DG2P or in Spark or DBD or CSNDD, which is an amazing database. You can go to GNOMAD through the website. You can, if your gene is involved in autism, you can see in the Safari. You can see where your gene is expressed. You can see the evolution of the gene. I have no time. If you're super motivated, you can adopt gene. I'm the father of SHANK1, SHANK2, SHANK3, a bad father, I have to say, because I didn't put so much information, but I will do it, Bert. And then you can also download everything. But you can see, for example, the odd ratio that I showed you. So this one is SCN2A here. This one is ASH1L, so you have the autism ratio, and this one is, so you can click, you can go on the website. You can ask for other information, and you can download everything, so you can play with the data on your computer. Last, a bonus track. This is what we do in the lab to try to after the genes. So these are proteins, synaptic scaffolding proteins. And SHANK3 was found, I showed you, but after we found SHANK1 and SHANK2 mutation, this is the IQ of the individuals. And this is the autism diagnostic interview. So the more you score, the more impaired you are in social interaction, verbal communication, nonverbal communication, or repetitive behavior. So these individuals with SHANK1, they have IQ like the general population. SHANK2 are intermediate, and patients with SHANK3, unfortunately, are very, very low IQ. So we went back to the patients. We could see some difference of clinical. I don't have time, but it's really important to go back to the patients. And so we met some mice. So we are super interested in why you are motivated to see the others and so on. And So this is an old story, but I will show you this. This is, on the left, it's a Sheng2 male, and we will introduce a female in estrus in the cage. And this is a Sheng2 mutant mice. So if you have never seen a mouse, a male and female mice in a cage, this is beautiful. So I hope you see the difference. We made the quantification, of course. So you see, he will be interested. But so 10 years ago, when we found that, we were very surprised because, I mean, you never know. I mean, what a mouse can do, and so we really wanted to understand the social interaction. So this is a microphone here, and so you see, it will follow her, but no, different. And so we were recording mice. They can sing in the ultrasonic vocalization. And I said to Elodie, if you want to know what these mice are saying, you need a lot of data. You need a massive, it was before AI stuff. And I said, if you want really to look at this, so we need like a YouTube for mice, like MouseTube. And so normally when I say stupid things like this, they don't care, but she made MouseTube. And so it's the first data sharing platform on mouse vocalization, if you're interested. But Elodie, she really wanted to see what's going on in the mouse. So it took us 8 years to do that. But now you can have your mice, and everything that they are doing, approach, contact, everything is in the database. You can really try to understand the complexity of the social interaction in mice. And it can be a super rich environment with Lego and so on. So we really wanted to see what it is to be a mouse, so we can so that you can play with exactly how they are. So we made this IKEA page, and then you can build. It's open source. You can build your system. Fabrice can answer all your questions, and AOD too. There are some colleagues who are using this for Fragile X and for other aspects. So now we have this complexity, so you can monitor them during 3 days and 3 nights, and you can record them. So we are now subtitling the movies and trying to understand what they're doing. So first message is that we put 2 males together and 2 females together. And yeah, so some people are, and when they vocalize, and really, so we are not working on males anymore. This is at 5 weeks, 3 months, 7 months, and girls. And more interestingly, the boys, when they vocalize, they are alone. And girls, when they vocalize, they are in group. So it's totally different, totally. And you don't have, I mean, the p-value, I mean, you don't have to. So, and the vocalization in girls are super complex. And really, we are interested to see what's going on. So we are doing new tests. This is a new test that I think it's really important for social motivation. So this is a test where the mouse can see, she can poke, open the door, and she has a choice to go to see other mice or to drink sugar water. So the first phase, it's the doors are open. Then the second phase, she can poke on the left or right, it's okay. Then only the left can open the door. Then they have to poke twice, then 4 times, then 6 times, then so they have to be super motivated. And then we'll go back to 1, and we reverse, so now it's the right poke to open the door. So the wild-type mice, they can do that, almost the 6th phase in 2 days. And it's interesting. They are more interested by the others instead of sugar water. I mean, maybe if we put more sugar, there will be. But okay, so they can do that, and then they are super motivated. They can really poke a lot of time to go to the, and they are a little bit more motivated by social than by sugar water. And the reversal learning phase is okay. So we looked at Sheng3. And Sheng3, we were very depressed because we made this super great, but but the shank-free heterozygotes was like this, and shank-free, the mouse couldn't open the door almost. So that was why doing this test for. But still, there are some resilient, so we are working on this. For SHANK2, it's super interesting, I think, that this is the, so they can do the test, but really, this is a time in social compartment. They are much less interested in the others. They are much less motivated by looking at the others. And something that we were surprised that we were looking at these mice, and most of them were going in the door. And they spent a lot of time in the door. And so we are working with autistic individuals now. So, there was a woman, an autistic woman who came to the lab, and she said, "But Thomas, you should have asked me. This is the best place to be in the test." And that's this place where you can be. And yeah, so that's something that we have to learn together. So we met, really, because I'm super late probably, but we met some IPS cells and of these shank-free patients, and we found that lithium can boost shank-free. This is something that for the haploid efficiency. And Gina Torrigiano in US, she found that she can reduce the grooming of these mice and restore the synaptic plasticity. I think it's an amazing period to be a mouse because you can be cured from everything, but humans are a bit more complex. So we made this clinical trials, and the last patient was included a couple of weeks ago. So we will see if lithium can help, because these patients have a really strong regression. Okay, are there genes in the human brain? Yes, but it's really an orchestra with some different effect. And this is my hero, and I always love that quote. I accept chaos. I'm not sure whether it accepts me. And I think if you work on autism or probably anything, but yeah. Okay. And so last slide, KB Brooks, she's a person with autism, and I like the way she illustrates this. She say, it's funny because when I talk with medical doctors, they see my life like this, and it's funny because I see my life like this, and I really like this idea. Okay, so the gene for something, it's probably more complex. So no mind left behind. I like this father who discovered the autistic traits of his daughter, and on the T-shirt it's written, I'm unique just like everyone else, and merci beaucoup.
Thank you. Question for Thomas. I will start. So you ended up with trajectories. So did you look at trajectories of some of your autistic patient and individual, and can you make some assumption about why they ended up with F1, F2, F3 of
No. When I started, so I'm an old researcher now, but when I started, it was autism yes or no with an Excel file with 50 patients. So now it's better. We have the SCQ, we have RBS, but at what time? I mean, no trajectories, nothing. So really, we need to get a better phenotyping and on longitudinal, for sure. So brief answer, no, I don't know. I have some ideas, of course, but this is a key question.
I have one while we're waiting for the room. So I thought it was great that you were able to use 23andMe questionnaires to get empathy, but we also know from UK Biobank that responsiveness is related to whether actually carrying loss of function variants and constrained genes. So it's sort of it's self-selecting out when you email people and ask them to do that. Yeah. So what biases are you afraid might creep in by doing kind of a post hoc questionnaire?
Yeah, I mean, there is huge bias. For example, the empathy quotient, the score in the 23andMe customers were the same as the other, but systemizing quotient, they were really quite higher. I mean, that was a long time ago, 5 years ago. So people were doing 23andMes. They are probably a little bit interested in what's going on with their genomes, or a little bit in genotype, probably. Yeah. super biased.
Pierre Betoun from Paris. Thanks for a wonderful overview. Are you looking at environmental effects like endocrine perturbators and metabolism around the thyroid hormone, fetal thyroid development? Because that could be a key factor in the increasing prevalence in the last 50 years.
So the brief answer is no, for one reason that if you have the data to show that these factors can increase the number of people with autism, I never seen the data on that aspect. But we are looking at a lot of environmental factors. A preterm birth, this is something that we are looking at, these preterm babies were excluded from the genetic studies because they already had one risk, which is, okay. So we are looking at these individuals. We are looking at socio-economic status. That is, super important. Some of these kids, the father left, because, and so what is the effect of the familial thing? So these are the environmental factors. I can list a lot, but the pollutions and perturbation and endocrine, it's another aspect. I will be happy to see what's going on, but if you have the data, show me the data, but I didn't see the data. In France, if you're a geneticist, you're right wing. If you work on the environment, you're left wing. So this is bad for me. So only one question from a French guy. Okay.